I'm honored to share our research with you in this webinar. My name is Changjun Yin. I'm a group leader in the Institute for Cardiovascular Prevention at the Ludwig-Maximilian University, Munich, Germany. The title of my presentation is ApoE attenuates unresolvable inflammation by complex formation with activated C1q. Here I briefly introduce Alzheimer's disease, in brief AD, as it relates to our research. AD is an aging-related disease and the major form of dementia worldwide. This pie chart shows the epidemiology of AD in the United States per year. It's apparent that few patients with AD are less than 65 years old. This group of AD patients is likely to carry one or more genetic mutations, such as APP or PS1 genes. Those cases fall into the category of early onset AD. In early onset AD, the A-beta hypothesis may explain mechanisms of disease pathogenesis. However, the large majority of AD patients are more than 65 years old and fall into the category of late onset AD. In this major group of AD patients, the mechanisms of disease pathogenesis is much more complex. Importantly, all the clinical trials targeting the A-beta hypothesis have not yet any promising result, putting the A-beta hypothesis into question. Those failures and the increasing data that late onset AD is a multiple factor disease, other than the traditional view, is forcing the research community to look for hypotheses that go beyond the A-beta hypothesis. Many alternative hypotheses have been proposed, including the role of blood-brain barrier, peripheral brain interactions, and many more. Here is one example. Blood-brain barrier breakdown is characterized by endothelial cell and parasite degenerations. Blood-brain barrier breakdown leads to accumulation of neurotoxic factors within brain, inflammatory responses and immune responses within brain, and others. Those changes trigger neural injury, synaptic dysfunction, loss of brain connectivities, which leads to neurodegenerative diseases. Amyloid beta plaques and neurofibular tangles are two major pathological hallmarks of AD. Here is a schematic figure showing normal brain and the brain with AD. Recent studies suggested the possibility that neuroinflammation may be a key to understand the molecular mechanisms of later onset AD. This is the single-cell transcriptome of brain microglia cells, which reveal disease-associated microglia cells. The immunofluorescence staining revealed a beta plaque-associated microglia cells. The second disease we are interested in is atherosclerosis. Importantly, atherosclerosis resembles AD in many respects. This phenomenon has not received sufficient attention in the past, in our view. Similar to late onset AD, atherosclerosis is also an aging-dependent disease whose pathogenesis is poorly understood. According to the World Health Organization's report in 2017, cardiovascular diseases are the leading cause of death worldwide. This figure delineates major aspects of atherosclerosis pathology, which begins in the inner layer of the arterial wall. Early atherosclerotic plaques form underneath the endothelial layer of the large and the middle-sized arteries. Clinical events occur in the advanced stages when plaque rupture releases its constituents into the bloodstream to circulation. Thrombi form to clog the circulating blood, triggering heart attacks, strokes, and other forms of tissue infarcts. The chronic inflammatory response hypothesis of atherosclerosis has received major attention in the recent years. According to this hypothesis, various immune cells participate and contribute to all stages of atherosclerosis. Although atherosclerosis and AD showed distinct pathological features, that is, lipid-rich plaques in the artery versus A-beta plaques in the brain, the large majority of aged individuals with dementia have both AD and vascular pathologists. In a landmark study in 1997, Hoffman and his colleagues examined 284 dementia patients and 1,698 individuals who were not demented. The results are shown here. The x-axis shows the peripheral atherosclerosis score measured by ultrasound. The y-axis is the percentages of people with Alzheimer's disease. This study showed there is a major synergism of peripheral atherosclerosis and AD, and this synergism is more pronounced in ApoE4 isoform carrier. The presence of shared risk factors of atherosclerosis and AD suggests that atherosclerosis and AD may also share common mechanisms at the level of risk factors for both diseases, including aging, ApoE4 isoform, hypertension, obesity, and hyperlipidemia. When considering the pathologies of each atherosclerosis and AD, we notice that ApoE protein is abundantly present in AD plaques in the brain and in atherosclerotic plaque. Here are ApoE protein staining in human brain with AD and ApoE staining in human artery with atherosclerosis, as shown by previous studies by others. ApoE is a multiple function protein harboring an N-terminal low-density lipoprotein receptor binding site and a C-terminal lipid binding region. 3 alleles have been reported in humans, that is ApoE2, ApoE3, and ApoE4. ApoE3 is considered the normal isoform. In 1993, Calder and his colleagues published several landmark studies showing that ApoE4 is the strongest genetic risk factor for late onset AD. The x-axis is age of onset of the disease. The y-axis is the estimated onset distribution. This line is non-ApoE4 carriers. This line is the individuals with 1 copy of ApoE4. This line is the individuals with 2 copies of ApoE4. This data showed that the individuals with 1 copy of ApoE4 will develop AD 10 years earlier when compared to non-ApoE4 carrier group. With 2 copies will develop AD 20 years earlier, indicating a strong gene dose effect of ApoE4 on AD development. Following this data, worldwide research races to uncover the underlying mechanism of the mysterious effect of ApoE on AD. Subsequently, different mechanisms were proposed to explain ApoE4 phenotype, including isoform-specific domain-domain interactions, different binding ability to lipoprotein receptors, and the maintenance of the blood-brain barrier. Before I show our own experimental data to you, I want to introduce the choroid plexus as a major brain structure that could be important in AD pathogenesis. Indeed, our data indicate, as you will see shortly, that choroid plexus may be a crucial interface between AD and atherosclerosis and may hold a key to understand the common mechanisms of the 2 diseases. Here is a schematic view of a brain. Here is brain parenchyma. The red is blood vessels. Blue is cerebrospinal fluid. The choroid plexus is a blood vessel-rich organ to produce the cerebrospinal fluid. The tidal junction is located between epithelial cells, forming the blood CSF barrier. This barrier protects the brain from molecules present in the circulation, while it expresses multiple carriers to regulate the interactions between the circulation and the brain parenchyma. We examined fresh frozen human choroid plexus using oil red O staining, which stains lipids in red. We searched for choroid plexus lipids in 30 human brains with various degrees of AD-associated pathologies. 17 cases were diagnosed with dementia and AD pathologists, and 13 cases were diagnosed as non-dementia with or without AD pathologists. We observed that all cases showed various degrees of lipid deposits in the choroid plexus. Importantly, we observed that lipid burden in the choroid plexus is much higher in dementia cases when compared to non-dementia cases, and the lipid burden associated with AD pathologists, including Bragg stages, A beta plaques measured by tau phases. Choroid plexus lipid burden is also related positively with the ApoE4 isoform. Those data indicated that choroid plexus lipid deposits may represent a new AD-associated pathology. In order to understand the molecular mechanisms behind those new pathologies, mouse models were used. We examined 6 aged groups of mice with distinct genotypes of either ApoE deficiency and the 2 humanized ApoE3 or ApoE4 knock-in mice on normal child diet or on a high-fat diet. They were around 1.5 years old. ApoE deficient and ApoE4 knock-in mice fed with high-fat diet developed lipid deposits in the choroid plexus, but wild-type mice and ApoE3 knock-in mice on high-fat diet did not. But it was not immediately clear why there was a strong isoform-specific ApoE4 effect. What seemed to be clear, however, was that hyperlipidemia seemed to be required to lead to lipid deposits. One way to understand the molecular mechanisms, transcriptome microarray analysis of choroid plexus may shed light on aspects of mechanisms in the different genotype choroid plexus. Those data showed 2-group comparison as indicated in the figure. The upper part of the pie chart represents upregulated genes in the mouse model in the upper part of the slide. The lower part of the pie chart represents the upregulated genes in the mouse model below. For example, when we look at the comparison of aged ApoE4 mice fed with child diet versus aged ApoE3 mice fed with child diet, 71% of differentially expressed genes, or in short, DEGs, are upregulated in ApoE4 mice when compared to ApoE3 mice. 29% of DEGs are upregulated in ApoE3 mice when compared to ApoE4 mice. Strikingly, among the upregulated genes in ApoE4 mice, 64% genes belongs to interferon-related genes. Similar observation can be found in ApoE4 mice fed with high-fat diet. Those data lead to 2 conclusions. First, there are isoform-specific gene expressions in the choroid plexus, and second, those gene expression patterns are modified by hyperlipidemia. The biological activities of the interferon-related genes range from regulation of autoimmunity by macrophages and dendritic cells to blood-brain barrier integrity, and more recently, distinct interferons have been found to modulate gamma secretase in Alzheimer's disease. Here, I show few examples of interferon-related genes, including interferon-induced protein 44, receptor transport protein 4, interferon regulator factor 7. Those data suggested that Those data suggest that choroid plexus interferon signatures are ApoE4 isoform dependent, and it is independent of hyperlipidemia. While these data are striking, more work is needed to understand each differentially regulated gene on choroid plexus homeostasis and its effect on brain biology and, of course, AD. Using the pan-leukocyte marker CD45, we observed leukocyte infiltration in the choroid plexus of ApoE knockout mice adjacent to lipid deposits, raising an important question: how could lipid deposits trigger local inflammation? Oxidized lipids are known to activate complement. This leads us to examine complement activation in choroid plexus. We observed strong immunoglobulin deposits together with lipid in the choroid plexus in ApoE knockout mice. As immunoglobulin deposits are known to activate complement, we speculated that the classical complement cascade may link the lipid directly and functionally to a local inflammatory tissue reaction. What are complement cascades? Complement is an ancient system and a major biological system regulating innate immunity. Moreover, recently, complement cascades have been shown to bridge innate and adaptive immunity. There are three complement cascades: the classical complement cascade, the lectin complement cascade, and the alternative complement cascade. As we can see from this schematic figure of the classical complement cascade, immune complexes binds to the C1 complex to initiate the classical complement cascade, involving cleavage of complement C4 and complement C2 to form C3 convertase. The C3 convertase cleaves complement C3 into C3a and C3b, which in turn generate the C5 convertase. The C5 convertase cleaves C5 into C5a and C5b, which in turn forms the complement membrane attack complex, with the result of triggering cell lysis. The activated complement cascade well forms the anaphylatoxin C3a and C5a to recruit leukocyte to the injury site. Both the anaphylatoxins, as one of the most powerful chemotaxins for immune cells, and the membrane attack complex generate one of the most powerful inflammatory tissue reactions known. By immunofluorescent staining, we confirmed complement C4, C2, C3, and C5 protein deposits associated with lipid deposits. Those data support the hypothesis that choroid plexus lipid deposits are causally involved in complement activation. Our next question was whether this interpretation that inflammatory response can be attenuated significantly by blocking components of the complement activation cascade. In order to address this question, we decided to target complement C5. That is a common target for all three complement cascades in aged ApoE deficient mice. We choose liver-specific C5 siRNA technology to target complement C5 in vivo therapeutically. C5 siRNA treatment largely reduced serum C5 protein level by more than 95%. Serum of C5 deficient DBA2 mouse was used as a genetic control. We indeed observed that liver C5 siRNA treatment reduced choroid plexus inflammation in aged ApoE deficient mice. Those data supported our hypothesis that a lipid complement inflammation axis controls choroid plexus inflammation and possibly affects brain homeostasis. Surprisingly, we found there is much less complement constitute in ApoE4 fed with high-fat diet mice when compared to ApoE knockout mice, despite the fact that they showed similar amounts of lipid deposits in the choroid plexus. This discrepancy got our attention as it seemed to indicate that the expression of ApoE4 was having an anti-complement effect. Strikingly, we found there is no complement protein deposits in ApoE3 fed with high-fat diet mice, indicating again an isotype-specific biology in the choroid plexus. Those data raise its important possibility that ApoE, here is ApoE4 in the knock-in mouse, may inhibit complement cascade in vivo. Indeed, we observed all ApoE isoforms inhibit the activities of the classical complement cascade by high affinity binding C1q in vitro. In order to examine the inhibition capacity of ApoE protein in vitro, we used the red blood cell hemolysis assays. In this assay, the measurement of hemolysis of the red blood cells reflect complement activities. When adding different amounts of ApoE proteins to the assay buffer, hemolysis of the red blood cells, while the classical complement cascade, are markedly reduced when compared to BSA control. All ApoE isoform inhibits the classical complement cascade, but not the alternative complement cascade. By various protein-protein interaction assays, we showed ApoE binds to C1q. Here are the results obtained by bilayer interferometry. Those data showed that all ApoE isoform binds to C1 complex. The C1 complex contains C1q, C1s, and C1r. Our result revealed that all ApoE isoform specifically binds to C1q, but not C1s and the C1r protein. By bilayer interferometry, we determined the binding affinity of all recombinant ApoE protein and serum-derived ApoE3, which is lipidated ApoE. The binding affinities of ApoE isoform to C1q range from 0.14 nanomolar to 0.58 nanomolar, which qualify as high affinity binding. As the positive control, the binding affinity of C1q, C1s is 2.32 nanomolar. When native C1q binds its initiators, like the immune complexes, native C1q changes its conformation to the activated C1q. This conformation changes is calcium dependent. We observed that the binding of ApoE with C1q is calcium dependent. We observed that the binding of ApoE with C1q is calcium dependent, which indicates that ApoE only binds the activated C1q, but not the native C1q. Using peptide assays, we observed that the binding region of ApoE to C1q is located between amino acid 139 to 152 of ApoE. Those data demonstrated that, indeed, all ApoE isoform inhibit the classical complement cascade, while complex formation with activated C1q in vitro. Our next question was, do C1q ApoE complexes occur in vivo? In order to detect protein-protein interaction in vivo, we used the proximity ligation assay. In the PLA assay, two primary antibodies were used to label two proteins in vivo in tissue sections. Two complementary nucleic acids were used to label two secondary antibodies. If two proteins are close, for example, less than 30 nanometer, the minus and minus sequences will bind to each other, which allows to be amplified and labeled by fluorescent signals. And those fluorescent signals can be detected by confocal microscopy. Therefore, PLA assay signal indicate protein-protein interaction in vivo. Importantly, in human choroid plexus tissue section, we observed C1q ApoE complexes in the choroid plexus deposition site. As PLA is a sensitive assay, we performed various control stainings. For example, C1q antibody alone, ApoE antibody alone, or no primary antibodies yield negative staining result. Those data revealed that the in vitro data can be translated to human brain as C1q ApoE complexes are enriched at the choroid plexus. Let me briefly summarize our data obtained in the choroid plexus. Our in vitro and human translational studies support the hypothesis that ApoE attenuates the classical complement cascade in vitro as well as in vivo in chronically inflamed human tissues. By genetic knockout experiment, we observed strong complement activation and inflammation in ApoE deficient choroid plexus, but to a much less degree in ApoE4 sufficient mice, indicating that knock-in human ApoE3 or ApoE4 rescued complement phenotypes seen in ApoE deficient mice. By C5 inhibition experiment in ApoE deficient mice, we were able to therapeutically rescue the inflammation phenotypes. All this data together support the hypothesis that ApoE attenuates choroid plexus inflammation by complex formation with C1q. Our next question was, do those mechanisms occur in other chronic inflammatory diseases, for example, in AD? ApoE protein deposition has been reported in A beta plaque before. Here, we confirmed this observation by performing co-localization of A beta plaque and ApoE protein both in a human brain and in APP-PS1 mouse brains. We therefore asked, does ApoE attenuate neuroinflammation by complex formation with activated C1q in Alzheimer's disease? Again, by using the PLA assay, we indeed observed that C1q ApoE complexes formed both in APP-PS1 mice as well as in human AD plaques. Here is a movie showed C1q ApoE complexes, the red dots, within and surrounding a beta plaque of a mouse brain with a beta plaque. In order to obtain evidence for the notion that A beta plaques activate complement cascade and promote neuroinflammation, we performed C5 siRNA treatment in APP-PS1-21 mice. Liver-specific C5 siRNA significantly reduced A beta plaque-associated microglial cells aggregation. By targeting liver C5 protein, we were able to modulate brain neuroinflammation. Those data indicate a presumptive liver-brain axis. How may this work? By ELISA, we confirmed the serum C5 protein level was markedly reduced. By high-resolution confocal imaging in 3D, we observed that serum immunoglobulins located outside of brain vessels and co-localized with A beta plaques. Those data were consistent with the notion that serum C5 protein contributes to A beta plaque-triggered neuroinflammation while a compromised blood-brain barrier in APP-PS1-21 mice. Moreover, targeting liver C5 is effective in modulating neuroinflammation. Those data reveal that ApoE attenuates neuroinflammation by complex formation with activated C1q in Alzheimer's disease. We showed at the beginning that ApoE protein deposits has been observed in human atherosclerotic plaque. Our next question was whether C1q ApoE complexes can be observed in atherosclerosis. By PLA assays, we observed C1q ApoE complexes in human atherosclerotic plaques. By high-resolution confocal microscopy, we observed that oxidized epitopes located on the surface of lipid deposits, which can initiate the complement cascades. We asked further, does ApoE also attenuate atherosclerosis inflammation by complex formation with activated C1q in atherosclerosis? C1q ApoE complexes that we observed in plaques indicated ongoing complement activation in human atherosclerotic plaques. We hypothesized that lipid complement inflammation controls atherosclerosis inflammation and plaque development. In order to address this hypothesis, we performed multiple experiments, including liver-specific C5 siRNA treatment in ApoE deficient mice. Our data show that liver-specific C5 siRNA largely reduced serum C5 protein level and reduced by 70% atherosclerotic plaque size in ApoE deficient mice. Those data support the conclusion that ApoE attenuates atherosclerosis inflammation by complex formation with activated C1q. To summarize, different initiators in different diseases binds to C1q complex to initiate the classical complement cascade, including immune complexes, oxidized lipids, and amyloid fibers. The activated complement cascade contributes to inflammatory tissue responses observed in atherosclerosis, choroid plexus lipid deposits, and Alzheimer's disease. Our data show that ApoE qualifies as a checkpoint inhibitor of the classical complement cascade. Complement-triggered inflammation is a common mechanism of atherosclerosis, Alzheimer's disease, and choroid plexus lipid deposits. Complement inhibition attenuates choroid plexus inflammation, a beta-associated inflammation, and atherosclerosis in mouse models. Choroid plexus lipid deposits may represent a new unrecognized hallmark of Alzheimer's disease. Here we studied 3 chronic inflammatory tissues, which is atherosclerosis, Alzheimer's disease, and inflamed choroid plexus. Choroid plexus lipid deposits show strong association with peripheral atherosclerosis and also show strong association with AD pathologies in brain. Mechanistically, we showed that aberrant immune responses are the common mechanisms of all 3 diseases, and complement inhibition inhibits inflammation in all 3 diseases in mice. Therefore, we proposed the interface model of atherosclerosis and AD. A better understanding of this model requires new approaches to investigate immune pathways in atherosclerosis and AD, and I believe that further studies of this model will yield new diagnostic tools and therapeutic targets for both diseases. I want to thank all our group members and collaborators. Without them, those studies could not have been possible. Particularly, I want to thank Andreas Habenich for his long-term mentorship. And more importantly, Andreas set an example for me of what a pure scientist should be. If you have any questions, feel free to contact me.